So I hope that a few of you have learned some things this morning about their objective, and some who have the right objective now know why, hopefully. So we're going to add a new layer of this and talk about refractive index mismatch. Difficult to say. Yes, and I think it is one, isn't it? Yes, okay. So I start. Right, so optical aberrations. So I spoke a little bit about optical aberration and refractive index mismatch, but now we're going to see exactly what it is. So this is one of the slides that I had for the objective, the specifications of the objectives. One of the specifications that is important is the immersion medium. Which medium do we put on the objective, right? This is what the immersion medium is. So we immerse the objective in some medium. That's why it's called that way. So the medium on the objective lens can be air, water, glycerol, silicone, etc. And it has a certain refractive index, whereas our sample has a different and likely heterogeneous refractive index. And so it's important to match them, the refractive index of the medium and of the objective. So why is that and what happens? So this is a picture of a sample that I have in the lab, which is just a plastic bottle with some marbles inside. And these are beads that are made of some sort of gel. You know, it's the type of things that you put in if you have like flowers, a flower pot that is transparent, and you fill this up with these type of beads. They absorb water, so they have the same refractive index as water. And so what happens is that if you have so here we have water, but not the whole thing, not the whole bottle is full of water. So we can see the beads here. These beads are in the air, whereas these beads are in water. And you can see that the marbles are sort of floating because they are held by more of these water bead underneath. So there's a gel beads from plants expanded in water. And then we have two glass marbles. If we look at this from the side, what we're seeing, we see the marbles very clearly. And that is because if you think about the light that our eyes are recording, you can see that first it goes through the air, and then it goes through thin plastic, which is the bottle, and then it goes through the beads that are in water. So all of them have the same refractive index as water. So we have the beads full of water, and then we have the space between the beads that is full of water as well. So everything has the same refractive index. And then we have the glass, which is the marble. But if now we look at the bottle from the top, this is what we see. We can hardly see the marbles. And even though you can see that the green marble is quite close to the beads here, so closer to the surface that we're looking at, we can hardly see where they are there. If you didn't know that they were marbles, you would probably not notice. And now, what is the light path from there, from the top view? Now we go for the air, and then we go to thin plastic, and then we go again to air, because there's a lot of air there. And then we go inside this water bead. So we have again the refractive index of water. And then we go again to air and beads and air and beads. And then finally we get to the marble, which is made of glass. So there's a lot of refractive indices here. And you can imagine that this is a lot less. We have only two layers of beads here. This is a lot less refractive index in like surfaces or interfaces than if we had a sample. There, if we had a sample, every cell has a different refractive index at the membrane, in the nucleus, in the cytoplasm, and everything. So that's what happens to the light. Both the excitation light and the emission light are going through this process when we have different refractive indices. So this is a problem, and this is scattering everywhere. So we get optical aberrations, which can lead to the fact that we don't even see where the marbles are. So what are optical aberrations is basically the point spread function is distorted. The point spread function is what we are recording. This is the optical image that the microscope makes of our sample. And if it's not, if it's distorted, if we have optical aberrations, we can have things like this, for example. It's very, very distorted. So the PSF is distorted, and the PSF is going to be different at different focus as well. So it means that also the different colors can be focused at different Z levels. And we can have also a different focus at the center and at the periphery of the image. So lots of things can happen that lead to spherical aberrations and optical aberrations. When the optical aberrations are due to a problem with the refractive index mismatch, so we have light go through, both excitation and emission go through lots of different refractive index interfaces, what can we do about it and what are the consequences? So in here, we have a schematic of a sample, an objective here. So this is an inverted microscope. It's pointing upwards. And then here we have the front lens of the objective. This is an oil objective. So the front lens is made of glass. So the refractive index is 1.52. And here we put oil on the objective. And so it has the same refractive index. And here we put a cover slip. And afterwards, we have our sample. Our sample is made of fluorescent beads, like small fluorophores, in vector shield. So vector shield has the refractive index of 1.44. So here we have same refractive index, same different. Okay. So what are the consequences? When we are imaging this bead, which is stuck on the cover slip, this is why we keep on like hammering. You need to put your sample directly against the cover slip. When we're imaging this bead, fine, we get a nice point spread function. If we're imaging this bead that is a little bit further away inside the sample, it's further from the cover slip. Oh, trouble. And now we're talking about only 25 microns away from the cover slip, you know? But actually, this effect starts immediately. So I'm sure that people who have imaged at high resolution a monolayer of cells have noticed that the resolution and the contrast is much better when you're imaging close to the cover slip than if you're imaging the top of the cell. So we're talking about maybe 10 microns. So actually this effect starts immediately as soon as the light has to travel through the new medium. So here we have a distorted PSF, and if we continue here, we get an even more distorted PSF. So this is the effect of having refraction index mismatch. And in this sample, we're lucky because the sample has the same refractive index everywhere. But if it's a heterogeneous sample, which is what our samples really are, then it's even worse. So we're going to have a little workshop this afternoon with JJ Jinzhong, who will demonstrate this to you. And so one of the problems is that even though we have maybe an adjustment ring on our objective that we could maybe turn, the problem is that the point spread function is going to be different at different depths. So it's very difficult to find one position. It's impossible to find one position that works for all the positions. Now we could have an automated, something that moves the color that is automated, but anyway, it's going to be impossible to use this only. And if we have, because it grows exponentially, the distortion grows exponentially when we're imaging. Even if we have an automated correction color, it's going to be difficult to have something that works everywhere. So it is a problem to have refraction index mismatch. And again, it depends on your scientific question. It's not a problem all the time. It all depends on our scientific question. The more stringent your scientific question is, the more careful you have to be with your sample preparation. And if you are trying to detect small differences, for example, between your mutant and your wild type, if you're after detecting and measuring small differences, you have to be super careful with your sample preparation. So what exactly happens if we look at this? Not only we get a point spread function that is bizarre, so then clearly the resolution is decreasing, but also we have a scale distortion. So if you are after measuring distances of objects in Z, so for example, you want to measure the volume of the object, then you're in trouble. And let's have a look at this. So here again, we have our objective. This is the front lens. This is, we will see that we will compare different objectives. So this is whatever we need to put on the objective. Cover slip is 1.52 because it's glass. And here we have the cell. And so the cytoplasm is pretty much the same refractive index as water, so 1.33. And here we have our little fluorescent bead. So we have refraction in here. We have refraction in both directions, right? On the way when the excitation light comes in and when the emission light comes out. Because here we have a difference in refractive index. So let's have a look. This this little bead sends a fluorescent light in all directions, and some of the light goes into the objective. And so there we see with ray optics that when we reach this interface, we have refraction, and so the light direction is changed. And there is, as I said before, there is a little law that tells you how much it's changed, and that depends on the, it's proportional to the difference between the two refractive indices. You can see that here. So here we have this first angle, and here we have a second angle. So the light is bent with a certain difference in the angle. And here, what happens, if we have a water objective here, we're going to have the refractive index here, another one here, and then another one here. So what happens then? We have refraction here. But the objective refractive index is the same as the sample refractive index, only we have a cover slip in between. If you have done your job very well and you used a number 1.5 cover slip, which is 170 microns, it is very easy for objective manufacturers to calculate how much the ray is going to be shifted. And so they can compensate for this. So you can see in here that we have this angle A, because we have a certain difference in refractive indices. But because we have the same refractive index in the sample and in the objective mounting medium, we have the same angle here. And therefore, when you want to measure your distances in Z, this is the real position of the sample compared to the cover slip. So that is a certain distance between the cover slip and this object, which allows you to measure distances in Z. And here we're going to have the same distance that will be measured because the image is formed with the same angle. So fine, there we have the correct data when it comes to measuring distances in Z and therefore volumes, if you are after volumes. Now, let's look at an air objective. What happens if here we have air instead of water? Now we have a difference we have three refracted indices, so two interfaces. So now we have the same thing, the beads, we have the same thing exactly here, same thing here through the cover slip. And now, because we have a higher one refractive index here, the angle is bigger. And so when this is the real distance between the fluorophore and the cover slip, but what we are measuring is lower, okay? So the sample is going to look like it's compressed. So each fluorophore will have the same problem, but the problem will be different depending on how far they are from the cover slip. A fluorophore close to the cover slip will have zero problem, and a fluorophore far away from the cover slip will have this problem. So this is why the sample looks compressed. If now we look at an oil objective, and what happens is that we have here, same thing exactly, but because the oil on the objective has the same refractive index as glass, then it will just go through like this. And what we have here is this is the real distance, but now our image is elongated because so this is a longer distance. The image is formed at the wrong place because of refraction, okay? So this is exactly what happens when we have refractive index mismatch. If we have a match between the refractive index of the objective immersion medium and the refractive index of the sample, the angle is the same, and that can be calculated. And regardless of where in your sample this bead is, you will always get the same angle. But if you do not match, this is what happens, and we get an object that is either compressed or elongated. So how to know if it's compressed or elongated? This is a little trick. If you follow the emitted light, so we follow the light that goes from the sample to the objective, the refractive index, when the refractive index increases, for example, here in the sample is 1.33, here is 1.52, it increases. So then the distance in the sample also increases. We get an elongation. So when we follow the emitted light, if the refractive index increases, we get elongation. If the refractive index decreases, we get a flattening of the object. And the further away from the cover slip, the worse the elongation. So this is not a linear process at all. So in another way of showing this is like this. And a consequence of having this mismatch is that effectively we are lowering the numerical aperture of the objective. So here is a sample that is in water, so same as before. Now it's an upright microscope, but it's the same. Here we have the cover slip, and we've done our job. So this cover slip is 170 micron thickness. But here we're using an oil objective, here we use a water objective. So in this case, we have a match between the objective immersion medium and the sample. And here we have a mismatch. So if we follow the emission light, the refractive index is increasing, and therefore our our object is going to be elongated. So you can see in here what happens. The fluorophores that are close to the cover slip, there will be no distortion, and the fluorophores that are far away from the cover slip, even though this is our object, it will look like a much more elongated blob because these fluorophores are going to be the light is going to be distorted. So here we have an elongated sphere, and in this case, the image is moved, is formed somewhere, but all the dimensions are correct. So if you're measuring volumes and distances in Z, you absolutely want to be super careful with matching the refraction index. So here there's no distortion at the cover slip, and there is distortion far away from the cover slip, and that is different at different distances from the cover slip. And in here we have distortion like the cover slip, and here we have the same distortion away from the cover slip, so we're good. And clearly the cover slip is the correct thickness. If you have the wrong thickness here, you are introducing these aberrations yourself. So the cover slip is part of the design of the objective. Only choose 1.5, number 1.5 cover slips. And the refractive index mismatch lowers the effective numerical aperture. You can see in here that the cone of light, you remember the numerical aperture depends on the cone, or the angle of the cone of light that comes inside the objective. And because of refraction here, the cone of light is smaller. So it's exactly the same thing as using an objective that has a smaller numerical aperture. So maybe you're going like, okay, I want a better resolution. I'm using now an air objective. I'm using a water objective, but I'm going to choose this objective because it has a higher numerical aperture. But you don't pay attention that there's a mismatch in the refractive index. You're actually using an objective with a higher numerical aperture, but you're not getting that numerical aperture. Okay, so the effective numerical aperture is lower, whereas in here we get the full numerical aperture. And this is the reminder of what the numerical aperture is. It is related to the angle of the cone of light that comes into the sample. So if we reduce the cone, we reduce the numerical aperture. So we have a lower effective numerical aperture. In the case of a mismatch, we have aberrations, elongated volume, because the refractive index increases over the emission light. And then we have a lower effective NA, and therefore we have a lower resolution. And that's exactly the reason why we changed objective to an oil objective, because you wanted a higher resolution. So it's not very good. And in here we have no aberration, correct volume, full numerical aperture, and therefore full resolution. So when you image samples that are in PBS or in medium, like live samples, always use a water objective or an air objective, but not an oil objective, okay? You can use oil objectives with samples that have a higher refractive index. For example, if you have a mounting medium that has a high refractive index, this is where you should use. And this is why I asked you to figure out what is the refractive index of your mounting medium. So we're going to work with this today. So what happens if you have the wrong cover slip? you get also optical aberration just by changing the cover slip and using whatever you have in the lab, which is likely number one if nobody has paid attention. So here I will show you the images of the same sample that is acquired in two different objectives. The sample is on the slide, and it's an aqueous medium, so a water-based medium. So the user here was, oh, it's an aqueous medium, water-based, so I'm going to use a water objective. Unfortunately, on this microscope, it was an upright microscope, and we had a dipping lens. And the dipping lens is designed to have no cover slip, but that's what he used. It's an excellent objective, 20x NA1.0 water dipping lens, but it is designed to have no cover slip. So this is what, and he was careful. So he used cover slip number 1.5. And he was really very careful with all his settings and everything. We will talk about Nyquist sampling next week. But then when we changed objective and we used an objective that has like almost the same numerical aperture, just a little bit lower, with a cover slip, because this objective is not a dipping lens, so it's designed to have a cover slip. You can appreciate now the difference, the enormous difference in contrast, because here we have introduced some optical aberrations because we use a cover slip with an objective that should not have a cover slip. And again, you will only see the difference if you compare side by side, and you may continue imaging this for a long time without realizing that there is, that you could get better. So here I want to show you, just looking again at my time, bloop. Good. So here, this is a sample from an image of a sample of one of the students a few years ago, and she was imaging spinal cord. So this is a section, and then we have three colors, so blue, green, and red, and then it's mounted in prolonged gold. Which was autofluorescent. You cannot see in this image, but it was autofluorescent. We found this out during the workshop, and we will do all this with you during your workshop. And then the sample was on the slide, not on the cover slip, unfortunately. And she was using a 10x objective NA 0.45, so it's an air objective. And she was focusing on the red. And then can anyone tell me if there is something wrong with this image? It's difficult to see.
I don't know if it's by the image per se, but it seems like on the top left corner, the contrast is higher than on the bottom right. But I don't know if maybe that's because of the
Yeah,
mix.
you're right, but I know why. You know why? You can see it because let me show you the next image and then you will see. Now, can you see what the problem is?
Ah, yeah, the blue channel.
Yes. So the blue is out of focus. The reason why you could see here that it was not so focused because there's more nuclei here than here. So in the overlay image, it was easier for you to see that. But you can understand how difficult it is to see anything when you have the overlay in color. So please do not do that. When you display your things, you put the overlay in color and you put each channel in black and white. And you can see now how easy it is to see that there is a problem with the blue channel. And actually, she focused on the red, and the green was also a little bit out of focus, but a little bit, but the blue very clearly out of focus. And what is that? It's refractive index mismatch. The sample is on the slide. There's a thick layer of mounting medium. Then we put the cover slip. We focus on the red, and the other channels are focused on the wrong, on a different Z level. So if you now focus on the blue, you will see that the red is out of focus. Okay. So this is chromatic aberrations. The blue channel is out of focus. So put your sample on the cover slip if it's possible. So not on the slide, even if it's a tissue. If it's not possible because you have some nasty antigen retrieval, when you use paraffin sections, you need to do antigen retrieval very often. So if you have to boil your sample and put it through a nuclear reactor, then you have to put the sample on the slide because then they stick better. But then if that case, you put as little mounting medium as possible to avoid the fact that your sample, when you put a lot of mounting medium, literally what you're doing is that you're bringing your sample further away from the cover slip. And you have seen that the distortions are bigger and they increase with the distance to the cover slip. This is why we want our sample to be as close to the cover slip as possible to avoid chromatic aberrations and all the optical aberrations. And then clearly show the channels one by one in black and white so that people can actually see what you want them to see. Right, so here are a few conclusions. Not matching the objective and sample refractive indices or using the wrong cover slip thickness leads to increased optical aberrations, reduced brightness, reduced resolution, loss of contrast, incorrect Z measurements and volumes, and decreased effective numerical aperture of the objective. This is one of the major causes, like optical aberrations are one of the major causes of signal loss when you image thick sample. And when I say thick, again, at 10 microns, you already see the effect, okay? So it's not very thick. So both the excitation light and the emission light are affected. And there's no problem if your sample is extremely, extremely thin and stuck against the cover slip. There, then you will not notice the difference. And the aberrations only appear a few microns away from the cover slip. And the problem increases exponentially with depth as the sample gets away from the cover slip. No thick sandwich of mounting medium, please. Okay, so how to prevent optical aberrations and refractive index mismatch? We want to use the correct cover slips, 1.5 or 1.5H. If you have very demanding applications, you must have the 1.5H, more precise. Do not use cover slips with dipping objectives because they are designed to not use cover slips. And put your sample on the cover slip, not on the slide, if it's possible, or use very little mounting medium. And we will see all this with Gabi on Monday. And always adjust the correction color if the objective has one. And because otherwise you're introducing these optical aberrations yourself by using the wrong position of the correction color. If possible, match the sample and the objective refractive indices. If it's not possible, try to use an objective that has a lower refractive index than the sample so that you can use the full numerical aperture. So if anything, if you cannot match, use a lower, for example, if you have an oil sample, use a water, if you have a water sample, use an air objective, not an oil objective, okay? Don't have an objective that has a higher refractive index than the sample. And if the sample touches the glass and is very thin, any objective can use when the sample touches the glass, the fluorophores that touch the glass will not be affected by a refractive index mismatch because the light does not have to travel through the medium. So tissues are, it's highly variable, but we can say about 1.44 refractive indices. But clearly it's varied and it's heterogeneous. So mostly if you have a tissue section, an objective that is silicone or glycerol is best. And it is clearly very best if you clear your tissues. We will talk about clearing later in the course on Monday. And if you have live cells in medium, always use a water objective or an air objective, but you will get the best resolution with a water objective. They have higher NAs. So this is what I wanted to say for now.